Alzheimer's disease. How does beta amyloid affect the synapse? When released from cells in the brain, beta amyloid can bind to receptors, stimulating a range of responses. These include receptor endocytosis, improper enzyme activation, and ion channel opening, allowing an undesirable influx of calcium into the cell. How does this impact the cell? The role of the proteasome is to degrade unwanted proteins, which is necessary to allow normal cellular function. Endocytosed beta amyloid can dissociate from the receptor, disrupt the vesicle membrane, and prevent the proteasome from functioning. Microtubules are components of the cytoskeleton that are stabilized by the protein tau. Their role is to allow the movement of vesicles in the cell, some of which may hold neurotransmitters, for example acetylcholine. Activated enzymes will add multiple phosphate groups to tau, preventing it from binding to microtubules and causing them to break down. Mitochondria are the organelles responsible for producing the molecule of energy within the cell, ATP. A large inappropriate influx of calcium can prevent this synthesis, thus reducing the amount of ATP within the cell. How does this influence the synapse? First, we're going to look at a healthy synapse. The cell depolarizes due to an influx of sodium, which causes voltage-gated calcium channels to open. The influx of calcium is what allows the vesicles to fuse with the membrane, releasing the neurotransmitter, acetylcholine, into the synaptic cleft. The acetylcholine passes across the cleft and binds the acetylcholine receptor. This activates the receptor, causing it to open, allowing sodium to enter the cell and the action potential to continue to the next neuron. When this is complete, acetylcholine esterase breaks down the acetylcholine, preventing the overactivation of the postsynaptic cell. In Alzheimer's disease, fewer vesicles are packaged with acetylcholine, and even less can reach and fuse with the plasma membrane due to the loss of proteasomal, microtubule, and mitochondrial function. Lower concentrations of acetylcholine are broken down before the signal can be passed to the next cell. This reduction in cholinergic signaling explains the loss of memory formation seen in those with Alzheimer's disease. Knowing the mechanisms of Alzheimer's disease allowed the development of a class of drugs called acetylcholine esterase inhibitors. These work competitively or non-competitively, preventing the breakdown of acetylcholine and increasing the amount of time that it is present in the synaptic cleft. This allows acetylcholine to bind to the receptor and the action potential to continue.